chapter 4, hopefully before my battery dies. My charger is where the children are. Okay, mitochondria are the sites of cellular respiration, a metabolic process that uses oxygen to generate ATP or energy. Chloroplasts, found in plants and algae, are the sites of photosynthesis, and peroxisomes are oxidative organelles. Mitochondria and chloroplasts have similarities with bacteria. They are both enveloped by a double membrane. They contain free ribosomes and circular DNA molecules, and they grow and reproduce somewhat independently in cells. This led to Lynn Margulis's theory of an endosymbiosis. So the endosymbiont theory is that an early ancestor of eukaryotic cells engulfed a non-photosynthetic prokaryotic cell, which formed an endosymbiont relationship with its host. The host cell and endosymbiont merged into a single organism, a eukaryotic cell with a mitochondria. At least one of these cells may have taken up a photosynthetic prokaryote, becoming the ancestor of cells that contain chloroplasts. So basically you have this cell that engulfed a bacterial cell that was able to undergo cellular respiration. At the same time, this cell engulfed a photosynthetic prokaryote. So one of the prokaryotes became the mitochondria of a eukaryotic cell, and the other photosynthetic prokaryote became the chloroplast. Mitochondria are in nearly all eukaryotic cells. They have a smooth outer membrane and an inner membrane folded into cristae. The inner membrane creates two compartments, the intermembrane space and the mitochondrial matrix. Some metabolic steps of cellular respiration are catalyzed in the mitochondrial matrix. Cristae present a large surface area for enzymes that synthesize ATP. Remember those enzymes are called ATP synthase. So here is the structure of your mitochondria. We're going to talk more about that when we get to cellular energetics, photosynthesis and cellular respiration, chapter 6. Chloroplasts contain chlorophyll. It is the chlorophyll that absorbs sunlight and allows the chloroplast to undergo photosynthesis. Just going to get my charger real quick and block my children out. It's okay, they're with their dad. Okay. Try and finish this before we get interrupted, which is highly unlikely. Okay. Chloroplasts are found in leaves and other green organs of plants and in algae. Chloroplast structure includes thylakoids, which are membranous sacs stacked to form a granum. Stroma is the internal fluid found in the chloroplast. The chloroplast is one of a group of plant organelles called plastids. Peroxisomes are specialized metabolic compartments bounded by a single membrane. They produce hydrogen peroxide and convert it to water. Peroxisomes perform reactions with many different functions. So here is a peroxisome, a chloroplast, and a mitochondria under the microscope. The cytoskeleton is a network of fibers extending throughout the cytoplasm. It organizes the cell's structures and activities and anchors many organelles. Here's what it would look like under the microscope. The cytoskeleton helps to support the cell and maintain its shape. It interacts with motor proteins to produce motility. Inside the cell, vesicles and other organelles can walk along the tracks provided by the cytoskeleton. So here is a picture of a vesicle moving along a microtubule which requires ATP or energy. Three main types of fibers make up the cytoskeleton. Microtubules are the thickest. Microfilaments, also called actin filaments, are the thinnest, and intermediate filaments are fibers with diameters in a middle range. Here are many of the functions. You are not required to memorize these. But here are some examples. Functions of microtubules include shape and support the cell, guide movement of the organelles, separate chromosomes during cell division. In animal cells, microtubules grow out from a centrosome near the nucleus. The centrosome is a microtubule organizing center. The centrosome has a pair of centrioles, each with 9 triplets of microtubules arranged in a ring. Remember that centrioles are only in animal cells. Microtubules control the beating of cilia and flagella. Microtubule containing extensions projecting from some cells. So remember that flagella, for example, function in movement of, for example, bacterial cells. Cilia, those are like the hair-like projections, for example, that are found in cells of your respiratory system. We're not going to worry about this, or this, or this. Microfilaments are thin, solid rods built from molecules of globular actin subunits. The structural role of microfilaments is to bear tension, resisting pulling forces within the cell. Bundles of microfilaments make up the core of microvilli of intestinal cells. So microvilli serve to increase the surface area of the intestines in order to increase the efficiency of nutrient absorption from your digestive system into your blood, and consequently to your cells. Microfilaments that function in cellular motility interact with the motor protein myosin. Remember, actin and myosin are the filaments that make up your muscle cells, your muscle tissue. Intermediate filaments are larger than microfilaments, but smaller than microtubules. They support cell shape and fix organelles in place. Most cells synthesize and secrete materials that are external to the plasma membrane. These extracellular materials are involved in many cellular functions. The cell wall is one such example. The cell wall is an extracellular structure that distinguishes plant cells from animal cells. Prokaryotes, fungi, and some protists also have cell walls. The cell wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. Remember that animal cells, if they take up too much water, they can burst. But plant cells, because they have a cell wall, it prevents their cells from bursting. Plant cell walls are made of cellulose fibers embedded in other polysaccharides and proteins. Don't worry about the different parts of the cell wall, but you should know that plasmodesmata are channels between adjacent plant cells, and that molecules can move through the plasmodesmata, and the function of that would be to allow adjacent cells to basically interact with each other, communicate with each other. Animal cells lack cell walls but are covered by an elaborate extracellular matrix, which is made up of glycoproteins such as collagen, proteoglycans, and fibronectin. The extracellular matrix proteins bind to receptor proteins in the plasma membrane called integrins. So here is your plasma membrane. You can see that there are proteins on the inside of the cell that are bound to the cytoskeleton, and that there are molecules, proteins on the outside of the cell that are bound to the cytoskeleton, the extracellular matrix, rather. Neighboring cells in an animal or plant often adhere, interact, and communicate through direct physical contact. There are several types of intercellular junctions that facilitate this: plasmodesmata in plants, and then tight junctions, desmosomes, and gap junctions in animals. Plasmodesmata are channels that perforate plant cell walls. Through plasmodesmata, water and small solutes can pass from cell to cell. Animal cells have three main types of cell junctions: tight junctions, desmosomes, and gap junctions. You don't have to know the difference between those, just that they exist. Cellular functions arise from cellular order. For example, a macrophage's ability to destroy bacteria involves the whole cell, coordinating components such as the cytoskeleton, lysosomes, and plasma membrane. Okay, that concludes Chapter 4.